junk DNA. But gradually this position has begun to look less tenable for a whole host of reasons. Perhaps the most fundamental reason for the shift in emphasis is the sheer volume of junk DNA that our cells contain. One of the biggest shocks when the human genome sequence was completed in 2001 was the discovery that over 98% of the DNA in a human cell is junk. It doesn't code for any proteins. The Shakespeare analogy used above is in fact a simplification. In genome terms, the ratio of gibberish to text is about 4 times as high as shown. There are over 50 letters of junk for every 1 letter of sense. There are other ways of envisaging this. Let's imagine we visit a car factory, perhaps for something high-end like a Ferrari. We would be pretty surprised if for every 2 people who were building a shiny red sports car, there were another 98 who were sitting around doing nothing. This would be ridiculous. So why would it be reasonable in our genomes? Well, it's a very fair point that it's the imperfections in organisms that are often the strongest evidence for descent from common ancestors. We humans really don't need an appendix. This seems like taking imperfection rather too far. A much more likely scenario in our car factory would be that for every 2 people assembling a car, there are 98 others doing all the things that keep a business moving. Raising finance, keeping accounts, publicizing the product, processing the pensions, cleaning the toilets, selling the cars, etc. This is probably a much better model for the role of junk in our genome. We can think of proteins as the final endpoints required for life, but they will never be properly produced and coordinated without the junk. 2 people can build a car, but they can't maintain a company selling it, and certainly can't turn it into a powerful and financially successful brand. Similarly, there's no point having 98 people mopping the floors and staffing the showrooms if there's nothing to sell. The whole organization only works when all the components are in place, and so it is with our genomes. The other shock from the sequencing of the human genome was the realization that the extraordinary complexities of human anatomy, physiology, intelligence, and behavior cannot be explained by referring to the classical model of genes. In terms of numbers of genes that code for proteins, humans contain pretty much the same quantity, around 20,000, as simple microscopic worms. Even more remarkably, most of the genes in the worms have directly equivalent genes in humans. As researchers deepened their analysis of what differentiates humans from other organisms at the DNA level, it became apparent that genes could not provide the explanation. In fact, only one genetic factor generally scaled with complexity. The only genomic feature that increased in number as animals became more complicated were the regions of junk DNA. The more sophisticated an organism, the higher the percentage of junk DNA it contains. Only now are scientists really exploring the controversial idea that junk DNA may hold the key to evolutionary complexity. In some ways, the question raised by these data is pretty obvious. If junk DNA is so important, what is it actually doing? What is its role in a cell if it isn't coding for proteins? It's becoming apparent that junk DNA actually has a multiplicity of different functions, perhaps unsurprisingly given how much of it there is. Some of it forms specific structures in the chromosomes, the enormous molecules into which our DNA is packaged. This junk prevents our DNA from unraveling and becoming damaged. As we age, these regions decrease in size, finally declining below a critical minimum. After that, our genetic material becomes susceptible to potentially catastrophic rearrangements that can lead to cell death or cancers. Other structural regions of junk DNA act as anchor points, where chromosomes are shared equally between different daughter cells during cell division. The term daughter cell means any cell created by division of a parental cell. It doesn't imply that the cell is female. Yet others act as insulation regions, restricting gene expression to specific regions of chromosomes. But a great deal